It's not my fault, I'm not to blame. These ain't my sins, I broke my chains. There's more to do and I still want to live.
I feel your breath upon my neck, a soft caress as cold as death.
Cold as death.
I feel your heart beat in my soul, our futures bound, our bodies know. Your blood like wine, I want it in. Oh darling, get me drunk, invite me. It's not my fault, I'm not to blame. These ain't my sins, I broke my chains. There's more to do if I can only live. I can't go yet, don't let me die. I'll never stop until I'm done. But just tonight, maybe I'll rest in peace. I feel your breath upon my neck, a soft caress as cold as death. I hear your heart beat in my soul, our endings bound, our bodies know. I can't go yet,
Don't let me die,
I want to live.
my only one. There's more to do if we can only live. The clock won't stop and this is what we get.
Hello, and welcome back to our last chapter for the day. That being is chapter 10, which has to do with infection. This is a long chapter, and looking at it, I think we're going to be reading for a good maybe 2.5 hours or so. We're not reading all of it. There are sections where we're jumping. We're gonna run from, I want to say, I'm reading all through the emerging infection, microorganisms in humans and their dynamic relationship. Microorganisms, but I think there's portions here that we're gonna start like jumping over. So I know on my end it's gonna say read pages 933 to 983, and well, here, let's just take a quick pick here. 83. So once infectious parasites and protozoans kicks in, that's when we'll jump ahead to 1007, and we'll start to pick back up at HIV. And then that finishes around 1036. So pretty much, I think it'll cover all of HIV. So with that being said, I know our pages are not going to like line up perfectly, but close enough. And And whatever doesn't, doesn't. It's okay. So I've got plenty of water here. I stocked back up, had some dinner, so that we can finish this chapter up before we take our quiz and then go hit the gym. So without much further ado, let's go! For most of human history, infection was the primary disease related to the cause of death, including the bubonic pneumonic plague, Spanish influenza, and smallpox. Modern public health initiatives, vaccination programs, and the use of antibiotics have greatly progressed prevention and treatment of infectious diseases. As a result of these initiatives, naturally occurring smallpox have been eradicated from the globe. The last reported case was in 1975 in Somalia. Measles is almost eradicated in the Western Hemisphere, and many diseases such as tuberculosis and polio are on the decline. In the United States in 2014, influenza pneumonia was the 8th and septicemia was the 11th ranked cause of death in adults. Bacterial sepsis was the 7th leading cause of neonatal deaths. However, infectious diseases remain a significant threat to life in many parts of the world, particularly in low-income countries where prevention is less effective. In these countries, 5 of the top 10 causes of death are attributable to infection: lower respiratory tract, HIV, AIDS, diarrheal diseases, malaria, and tuberculosis. Infectious disease remains a significant cause of morbidity and mortality because of the emergence of previously unknown infections. The reemergence and spread of old infections that were thought to be under control and the development of infectious agents that are resistant to multiple antibiotics. The cause of these occurrences are numerous and include the following. Vast and rapid urbanization in many areas of the world, resulting in a breakout in public health programs and a more rapid spread of infection. Poverty and social inequality, war and famine, global travel, allowing for more rapid spread of disease from isolated areas to virtually any point around the world within a few hours. Human encroachment into wilderness areas, resulting in contact with previously sequestered infectious agents. Practice of prescribing antibiotics excessively, or not taking antibiotics for a complete course of therapy, or even when appropriately used, over-administration of antibiotics facilitates the emergence of antibiotic-resistant microorganisms. Denial of a problem by governments, allowing infections to spread in an uncontrolled way. Diminished use of effective insecticides, and increased global warming, allowing insect vectors to spread into the breed in areas that were previously too cool for them. So, emerging infections. The emergence of previously unknown infections is not a new event in human history. However, the current rate may be unprecedented. Within one generation, more than 40 previously unknown infections have arisen. Some of these infections were harbored in animals and spread to humans with significant clinical impact. Severe acute respiratory syndrome, or SARS from coronavirus from bats. Middle East respiratory syndrome, or MERS, from dromedary camels. These are referred to as zoonotic infections. The Zika virus was also an example of a recently emerging infection that has spread globally. Several have extreme high mortality rates of more than 50%, including SARS, in those older than 65. Ebola, Marburg virus or mad cow disease, the Nipah virus, and the acquired immunodeficiency syndrome. However, most have spread very slowly. or initially appears in relatively isolated areas and are effectively controlled via quarantine. Although none of these infections has developed into worldwide scourges, the potential of reversion to more rapidly spreading variants is a concern of public health agencies. In 2015, the World Health Organization developed a list of emerging viral diseases that need urgent attention, based on a probability of causing severe epidemics and a lack of medical countermeasures. These included MERS, SARS coronavirus, the Nipah virus, and hemorrhagic fevers associated with Ebola, Marburg virus, Lassa fever, Rift Valley fever, and Crimean Congo hemorrhagic fever. Keep in mind, this book was released in 2019, prior to the coronavirus. So, who knew that this very section of the chapter is semi-predicting what happened without it being, written out. So we're going to skip over the Zika virus. At least 20 previously known infections were re-emerging, including a new strain of cholera, malaria, yellow fever, dengue fever, diphtheria, plague, and Marburg virus. The incidence of tuberculosis has risen by almost 33% between the mid-1980s to early 1990s. Although the United States is relatively free of most of these diseases, the effects of global warming and the relaxed control of vectors may result in resurgences. The West Nile virus, spread by mosquitoes, has spread throughout the continental US, Canada, and Mexico. Many common infections previously controlled by antibiotics or vaccinations have reemerged because of antibiotic resistance or secondary to decreased compliance with recommended vaccinations. Examples of these include the Streptococcus pneumonia, Staphylococcus aureus, tuberculosis, diarrheal disease, malaria, meningitis, respiratory tract infections, STDs, and HIVs. Additionally, noncompliance with recommended vaccinations has led to outbreaks of disease that were once under control, like measles and pertussis. Added to this collage of microbiologic dangers is the rising risk of bioterrorism. Agents such as smallpox, anthrax, and plague are continuing threats to public health and safety. Microorganisms and humans, a dynamic relationship. For many microorganisms, the human body is a hospital site in which to grow and flourish because of sufficient nutrients and appropriate conditions of temperature and humidity. Frequently, a symbiotic relationship exists and both humans and microorganisms benefit. These microorganisms make up the normal microbiome, the resident microorganisms found in different parts of the body, including the skin, mouth, GI tract, respiratory tract, and genital tract. For instance, the normal bacterial microbiome of the human gut is provided with nutrients from ingested food, produces enzymes that facilitate the digestion and use of many of the more complex molecules found in the human diet, produces antibacterial factors, or bacteriocins and colicins that prevent colonization by pathogenic microorganisms and produces usable metabolites, vitamin K and B vitamins. This beneficial homeostasis is normally maintained through the physical integrity of the gut and other mechanisms that sequester these microorganisms on the mucosal surface. So, the benefit or having microorganisms and the relationship with humans. There is a symbiosis that benefit only the human, no harm to the microorganism. Mutualism benefits to both of us, the human and the microorganism. Commensalism benefits the microorganism but has no harm to the human. Then there is the pathogenicity that benefits the microorganism but harms the human. and opportunism, a situation in which benign microorganisms become pathogenic because of the decreased human host's resistance. So, again, microorganisms and infections. The clinical infectious disease. Microorganisms are generally classified based on different morphologic characteristics and life cycles. However, groups of disease-causing microorganisms share many properties related to the clinical disease, processes of infection, and evasion of human protective systems. The clinical process of infection occurs in the following four distinct stages. We have an incubation period from initial exposure to the infectious agent and the onset of the first symptom, during which the microorganism has entered the individual, undergone initial colonization, and begun multiplying, but are at insufficient numbers to cause symptoms. This period may last from several hours to even years. The prodromal stage, the occurrence of initial symptoms, which are often very mild and include feeling a discomfort or tiredness. The invasion period, the pathogen is multiplying rapidly, invading further and affecting the tissue at the site of initial colonization. As well as other areas, the immune and inflammatory responses have been triggered. Symptoms may be specifically related to the pathogen or to the inflammatory response. And then convalescence. In most instances, the individual's immune and inflammatory systems have successfully removed the infectious agent and symptoms decline. Alternatively, the disease may be fatal or may enter a latency phase with resolution of symptoms until reactivation at a later time. So, clinical manifestations of disease vary depending on the pathogen, the organ system affected, and the intensity of the inflammatory response. Disease may be clinical, measurable infection-related symptoms, or subclinical, no apparent symptoms although the individual is infected. Effects of infection may be acute or chronic, secondary to the immune or inflammatory response, or a direct consequence of toxins or injury to infected cells. The initial symptoms are typically fatigue, malaise, weakness, and loss of concentration. Generalized aching and loss of appetite are common complaints. However, the hallmark of most infectious diseases is fever. Fever is not failure of the body to regulate temperature, rather, body temperature is being regulated to a higher level than normal. The hypothalamus functions as a central thermostat and regulates temperature. Pyrogens are agents that produce fever. Those derived from outside the host are termed exogenous pyrogens, and those produced by the individual's inflammatory response are termed endogenous pyrogens. Exogenous pyrogens indirectly affect the hypothalamus through the release of endogenous pyrogens by the cell of the host. A number of cytokines have been identified as endogenous pyrogens, such as interleukin-1 and interleukin-6, interferon, tumor necrosis factor, and some others. These cytokines raise the thermoregulatory set point through stimulation and prostaglandin synthesis in both thermoregulatory brain and non-thermoregulatory peripheral tissue. Fever is considered a beneficial adaptive host defense response in infection. Killing of temperature-sensitive microorganisms. Several factors influence the capacity of a microorganism to cause disease. So, communicability, the ability to spread from one individual to others and cause disease, such as the measles or pertussis, spread very easily. HIV is of a lower communicability. Immunogenicity, the ability to induce an immune response. Infectivity, the ability to invade and multiply in the host. Mechanism of action is how the microorganism damages the tissue. Pathogenicity, the ability to produce disease success, depends on the communicability, infectivity, extent of tissue damage, and virulence. Portal of entry, the route by which the microorganism infects the host, whether it's direct contact, inhalation, ingestion, or bites of an animal or insect. Toxigenicity, the ability to produce soluble toxins or endotoxins, factor that greatly influence the degree of virulence. Virulence is the capacity to cause disease. For example, the measles virus is of low virulence. Rabies virus is highly virulent. Infectious disease also are classified by their prevalence and spread within the community. Endemic diseases with relatively high but constant rates of infection in a particular population. Epidemic, the number of new infections in a particular population greatly exceeds the number of usually observed. Or a pandemic, an epidemic that spread over a large area such as a continent or worldwide. So we definitely had a pandemic when we dealt with COVID-19. As a consequence of the relative severity of clinical infection and ability to successfully treat the infection, some infections are considered relatively minor inconveniences, such as the common cold, and others have a major impact because of severe morbidity and mortality. The CDC have developed a national notifiable disease surveillance system. An evolving list of notifiable infectious diseases is maintained to be monitored, controlled, and to prevent the spread of disease. STDs and STIs are the most common reportable diseases in 2015: chlamydia, gonorrhea, HIV, and syphilis. The process of infection includes colonization, invasion, multiplication, and dissemination of pathogenic microorganisms. Many potentially pathogenic microorganisms reside within the normal microbiome without causing severe disease. The symbiotic relationship with the normal flora is maintained by physical barriers, the skin and lining of the respiratory and intestinal tract. The complexity of the microbiome and the inflammatory and immune system. Microorganisms that cause infection of the protective barriers of or defense systems are weakened and are referred to as opportunistic microorganisms. Physical damage to the intestinal tract during trauma or surgery release intestinal bacteria into the bloodstream, potentially leading to sepsis, shock, and death. Cuts in the skin may allow normally contained bacteria, like Staphylococcus aureus, to cause local infections, abscesses, and boils, and invade further and infect various organs. Alterations in the microbiome by antibiotics may allow local overgrowth of opportunistic microorganisms, such as Clostridium difficile and Candida albicans. Immune deficiencies may allow invasive systemic infections, like systemic fungal infections. Unlike opportunistic infectious agents, true pathogens have devised means to circumvent the individual's defenses and cause infection. Infection with these agents is usually dependent on adequate numbers of microorganisms rather than the compromise of the host's defenses. The estimated minimum number of microorganisms needed to cause infection, or minimum infective dose, varies greatly with the particular pathogen. The Vibrio cholera, 10 to the third to 10 to the 8th microorganisms. The norovirus or rotavirus is 10 to 100. Giardia lamblia, the parasitic diarrhea, and Mycobacterium tuberculosis, less than 10. So, pathogenic microorganisms usually exist in reservoirs, a natural habitat where microorganisms can multiply. such as the environment, contaminated water, soil, vertebrate animals, or another human who is infected. An individual may obtain an infectious microorganism from a reservoir either directly or indirectly. Direct transmission may occur through direct contact with infectious or another individual, such as the skin lesions of impetigo or S. aureus streptococcus pyogenes. Fungal skin infections like athlete's foot, scabies, mites, lice, head or body lice, oral herpes, or STDs. Vertical transmission is the spread of microorganisms from mother to child across the placenta, like T. pallidum, Listeria monocytogenes, cytomegalovirus, Toxoplasma gondii, ascending the birth canal from the vaginal colonization or during delivery, like Group B Streptococcus, E. coli, C. trachomatis, gonorrhea, hepatitis B, HIV, C. albicans, or through breast milk, like Staph aureus and HIV. Spread of microorganisms from one person to another is horizontal transmission. Indirect transmission occurs from contact with contaminated material, which can range from towels to food or through a vector. Transmission by indirect physical contact may occur with materials that the infected individual came in contact with, towels, sheets, clothing, toys, bandages, and is a common route of transmission for S. aureus. Iatrogenic transmission occurs inadvertently as a result of medical procedures, such as contaminated transfusions, transplants, implants, and catheters. Many infectious microorganisms are transmitted on the surface of objects that penetrate the skin or from dirt that contaminates wounds, such as spores of Clostridium perfringens, gas gangrene, or C. tetani, tetanus. Respiratory transmissions may occur by means of several mechanisms. Coughing or sneezing generally creates a mist that can be inhaled from the air for considerable time. This mechanism readily transmits many viral diseases, including the common cold, influenza, mumps, and measles. Infected individuals may also produce airborne droplets that settle on environmental surfaces. Several viral and bacterial diseases may be spread in this manner, including pertussis, influenza, pneumonia, Legionnaire's disease, and rubella. Other microorganisms may be airborne by circulating the air that carries microorganisms picked up from the soil, dried droppings, or other sites. Most pathogenic fungi, Histoplasma capsulatum, coccidioides, and mites, Blastomyces dermatitis, grow as saprophytes in the environment and are transmitted by inhalation of spores and initially present as pulmonary infections, but may spread to other sites of the body. Many ingested substances, food and water, may be contaminated by minuscule amounts of human feces. The fecal-oral transmission that transmits salmonella, or food poisoning, cholera, hepatitis A virus, poliovirus, rotavirus, norovirus, Giardia lamblia, and many others that cause minor or major gastrointestinal diseases. Zoonotic infections may be directly transmitted from animals, such as transmissions of the rabies virus through bites. Most zoonotic infections are transmitted indirectly by means of vector-borne transmission, primarily insects. The mechanical vectors, like a housefly, passively transfers parasitic, bacterial, fungal, or viral microorganisms on the outside of their body from contaminated material to an individual. Biological vectors, such as insects, feed off of the blood of infected individuals. The infectious agent usually undergoes part of its life cycle within the arthropod, including multiplication, and is transmitted through bites or stings. Biologic vectors are responsible for some of the most severe blood-borne diseases. Important arthropods include mosquitoes that spread malaria, arbovirus, dengue, tsetse, or dengue hemorrhagic fever, and then the tsetse fly that spreads trypanosomes, such as Trypanosoma cruzi, which causes Chagas disease in South America, and T. brucei, which causes sleeping sickness in Africa. Fleas that can cause the plague or typhus, the Rickettsia prausachia, Leishmaniasis, the protozoan parasite, and the deer tick for Lyme's disease. During transmission, successful pathogens usually undergo initial adherence to tissue at the site of entrance to the body. Several produce localized infections without tissue invasion, whereas others require spread from the initial site of colonization. Colonization begins when the microorganism stabilizes adherence to tissue through specific surface receptors. Adherence helps protect the microorganism from removal by mechanical, nonspecific forces, such as coughing or the respiratory mucus. Adherence occurs between ligand receptors of the microorganism and on the surface of cells, the specificity of which results in localization of an infectious agent to particular sites. Tissue tropism, such as confinement of common cold viruses to the respiratory tract. Although discussion of infectious diseases generally focus on single microorganisms that can be isolated and identified, they frequently exist in the body as part of complex multicellular masses called biofilms. Biofilms consist of mixed species of microorganisms including bacteria and fungi immersed in a highly organized extracellular matrix produced by the microorganism. Growth in biofilms offers survival advantages of protection by trapping components of the host's defenses and antibiotics. The closely organized structure allows for exchange of genetic information for antibiotic resistance and other defense mechanisms. Biofilms form on implanted medical devices such as catheters, pacemakers, implanted heart valves, prosthetic joints, and dentures, and are associated with chronic infections, persistent nasopharyngeal colonization with staphylococci, otitis media, diabetes-associated foot ulcers, infected burns, vaginitis, osteomyelitis, pneumonia secondary to cystic fibrosis, diseases of the oral cavity related to dental plaque, such as dental caries and periodontitis. Invasion follows colonization. Successful infectious agents have developed mechanisms to penetrate the tissue and evade the host's nonspecific and specific defenses. Inflammation and immunity. The process of invasion is closely linked to tissue damage. Extracellular pathogens generally invade by means of direct extension into the surrounding tissue and access to the lymphatic and bloodstream, from which they spread to internal organs. This form of invasion requires a variety of virulence factors, including adhesion molecules and toxins, which may directly lead to tissue damage. Obligate intracellular pathogens, such as the intracellular bacteria, parasites, and viruses, spread directly from cell to cell or by release of infectious microorganisms into extracellular fluid or blood from which they infect another cell. Some intracellular microorganisms may enter a latency phase within infected cells without spread or cellular damage until reactivation of the microbe occurs, like herpes, CMV, or tuberculosis. The presence of infectious microorganisms in the blood is generally referred to as bacteremia. Bacteria in the blood viremia, viruses in the blood, or fungemia, fungi in blood, and is usually related to transport and spread to other organs. Septicemia is multiplication of microorganisms, particularly bacteria in blood. Classes of infectious microorganisms. Infectious disease can be caused by microorganisms that range in size from 20 nm, like the poliovirus, to 10 meters, a tapeworm. Classes of pathogenic microorganisms and their characteristics are summarized in Table 10.1 and discussed in detail in the following sections. So, we've got different types of bacteria. Oh, excuse me. Bacteria, chlamydia, rickets, mycoplasma, fungi, protozoa, helminths, and viruses. So, infectious bacteria. Bacteria are prokaryotic unicellular microorganisms with no nuclei, mitochondria, or membrane-bound organelles. They are generally divided into several groups. True bacteria divide by binary fission and may have a variety of morphologies, including cocci, bacilli, so cocci, spherical, bacilli, rod-shaped, vibrios, comma-shaped, or spirilla, twisted rod shapes. Most disease-causing bacteria fall into these classifications. Then there is a filamentous bacteria, which have branching mycelium-like structures that resemble fungi, such as Mycobacterium tuberculosis, and leprae that cause tuberculosis and leprosy. Spirochetes are flexible spiral filaments that are motile, and most are anaerobic, such as Borrelia recurrentis, relapsing fever, Trypanosoma pallidum, syphilis, or Borrelia burgdorferi, Lyme's disease. Mycoplasma lack a rigid cell wall and are small and pleomorphic. They are the smallest and simplest member of bacteria, so Mycoplasma pneumonia causes atypical pneumonia, and Mycoplasma genitalium is the suspected cause of urethritis and pelvic inflammatory disease. Rickettsia is a strict intracellular parasite that can be rod-shaped, spherical, or pleomorphic. They are typically spread by insect vectors that cause Rocky Mountain spotted fever and typhus. Chlamydia are strict intracellular parasites, but with more complex intracellular life cycles. The primary chlamydial pathogen is Chlamydia trachomatis, which causes the common bacterial STI, pelvic inflammatory disease, and eye infection, conjunctivitis. Bacteria can also be categorized as a gram-negative or gram-positive. Gram-negative bacteria do not retain crystal violet dye in their gram staining process, whereas gram-positive bacteria do retain crystal violet dye. Gram-negative bacteria also have a lipopolysaccharide that coat in an outer membrane, which consists of a lipid A, a core polysaccharide, and O antigen. The lipopolysaccharide coat also is known as endotoxin, and there's a common bacterial pathogens that are listed in Table 10.2. So yeah, there's actually quite a bit. So, the mechanism of bacterial invasion and tissue. Many bacteria have specialized surface structures that contribute to the structural integrity of biofilms and provide adherence to cells and tissue during invasion. Pili, also called fimbriae, are thin, rod-like projections from the bacterial surface. Pathogenic strains of E. coli involved in urinary tract infections have a variety of different specific pili or pili-associated adhesion molecules, or adhesins, including mannose binding proteins that bind with glycoproteins specifically expressed on the bladder epithelium. Flagella, used for bacterial motion, also express adhesins. The Vibrio cholera expresses adhesins for fibronectin, a common component of mucosal cell surfaces. Adhesion to connective tissue components such as fibronectin or S. pyogenes, A. streptococci, or S. aureus, T. pallidum, collagen, laminin, vitronectin are common sites of bacterial adherence. Several microorganisms use complement-related receptors for initial attachment, like the Neisseria spp, adhere to the urinary tract epithelial cell membrane associated with cofactor protein, CD46, which is a complement regulatory protein that protects cells by inactivating C3b and C4b. Others use different components of the CR3, C3b, C3bi receptor-ligand system on the surface of monocytes, macrophages, and adherents, including the Bordetella pertussis, Legionella pneumophila, and tuberculosis. Bacterial pathogens produce a variety of toxic molecules that may kill the individual cells, disrupt the tissue, and protect against inflammation. Exotoxins are proteins released during microbial growth, and they may have specific or broad effects on connective tissues and cells. Exotoxins are classified by their mode of action. Type 1 bind to the cell surface and alter cell function, whereas type 2 toxins cause membrane damage, and type 3 enter the cell. Type 1 toxins include superantigens and heat-stable enterotoxins, so toxins that target the intestinal tract. Superantigens, or SAGs, are molecules that react with major histocompatibility complexes, class II proteins on the surface of antigen-presenting cells and the T cell receptors. Independent of the specificity of the TCR, SAGs activate many more cells than a specific antigen would and lead to overproduction of pro-inflammatory cytokines, such as interleukin-1, interleukin-2, and tumor necrosis factor alpha. SAGs are responsible for several diseases, including food poisoning, so S. aureus and C. perfringens, toxic shock syndrome, such as S. aureus, and scarlet fever, such as S. pyogenes. E. coli secretes a small molecular weight heat-stable enterotoxin that binds to the membrane-bound guanylate cyclase, resulting in increased levels of cyclic guanosine monophosphate and loss of cellular electrolytes and water. Type 2 toxins are membrane-damaging molecules of several subtypes. Cholesterol-dependent toxins are cytolytic. They bind to the cell surface and assemble to construct an extremely large pore, similar to C9 and the complement system and perforin produced by T cytotoxic cells. Cholesterol is required for formation of the pore. The pore leads to rapid exchange of calcium and water and cell death. As pneumonia secretes the cholesterol-dependent toxin pneumolysin, which can facilitate colonization of the lungs as well as protect the bacteria from macrophages. Type 2 toxins also include a subtype called RTX cytotoxins, which contain repeats in protein sequence. An example is alpha-hemolysin from E. coli. The alpha-hemolysin is a virulence factor that predominantly causes urinary tract infection and lysis of erythrocytes as a source of nutrients, but also protects E. coli by lysis of macrophages and lymphocytes. A third subtype of type 2 toxins include enzymatically active toxins. C. perfringens, found in soil and as a wound contaminant, is the cause of gas gangrene. The major virulence factor in the alpha toxin with phospholipase activity that results in hemolysis, as well as hydrolysis of membrane phospholipids of other cells. Type 3 toxins are generally AB toxins and the most common bacterial toxin. AB toxins are referred to as binary toxins because two different components are required for activity, separately secreted A and B components. The B components are activated by proteases either in the extracellular fluid or on the surface membrane of the target cell. Multiple activated B components organize on the cell membrane to form a complex, with which A components bind for transportation into the cell. Overgrowth of C. difficile in the colon is a common complication of prolonged antibiotic therapy that kills much of the commensal microbiome. C. difficile secretes membrane-disrupting toxins A, an enterotoxin, and B, a cytotoxin, that damages the mucosa and results in watery diarrhea and potentially life-threatening pseudomembranous colitis. Clostridium botulinum produces the most potent toxin known, botulinum toxin, which is a paralytic neurotoxin that blocks the release of acetylcholine at nerve muscle synapses and results in flaccid paralysis and death. Examples of AB toxins include botulinum, tetanospasmin, so Corynebacterium diphtheria, and P. aeruginosa, and Bacillus anthracis. A subset of AB toxins is AB5 toxins, examples of which are B. pertussis, S. dysenteria, the Shiga toxin, and V. cholera, cholera heat label enterotoxin. AB5 toxins form a complex of a component non-covalent bound in a ring of 5 B components. During endocytosis, the A component is activated and alters cellular function, usually by inactivating signaling pathways, increasing intracellular cyclic adenosine monophosphate, or other means. The A component of anthrax AB toxin is a zinc metalloproteinase that prevents intracellular signaling. Diphtheria is a respiratory tract infection with systemic complications caused by an AB toxin that inhibits messenger RNA translation and causes low blood pressure, necrosis in the heart and liver, degeneration of myelin sheaths, and death. The AB5 cholera toxin leads to increased adenylate cyclase activity, which affects chloride channels, causing an efflux of chloride, water, and other salts into the intestinal lumen. The Shiga toxin, another AB5 toxin, binds to the carbohydrates on the cell surface of the endothelium of the intestine, kidney, and brain. Shiga toxin is secreted by several pathogens, including E. coli, a contaminant of food or water. Ingestion of the Shiga toxin may initially cause bloody diarrhea, followed by a hemolytic uremic syndrome, hemolytic anemia, acute kidney failure, thrombocytopenia, which is especially severe and fatal in children. Another type of toxin is endotoxin. Unlike secreted exotoxins, endotoxins also called lipopolysaccharides, is contained in the cell walls of gram-negative bacteria and is released during lysis, or destruction of the bacteria. The innermost part of the LPS lipid A is made of a polysaccharide and fatty acid and is responsible for the substance's toxic effect. Endotoxins also may be released during bacterial growth or during treatment with antibiotics. Therefore, antibiotics cannot prevent the effects of an endotoxin. The release of a sufficient amount of endotoxin can lead to fatal endotoxic shock or septic shock, which is one of the leading causes of death in intensive care units. Bacteria that produces endotoxins are called pyrogenic bacteria because they stimulate the release of inflammatory mediators that produce fever and cause the local and systemic effects of inflammation. Once in the blood, endotoxins cause the release of vasoactive peptides and cytokines that affect blood vessels, producing vasodilation, which reduces blood pressure, causes decreased oxygen delivery, and produces subsequent cardiovascular shock. Endotoxins can activate the coagulation cascade, leading to the syndrome of disseminated or diffuse intravascular coagulation. Additionally, endotoxins induce the release of tumor necrosing factor alpha by macrophages. TNF is a potent pro-inflammatory cytokine, also called cachectin, because of its role in promoting cachexia, or wasting syndrome, in individuals with cancer. Cachexia is discussed in Chapter 12. Cytokines are discussed in Chapters 7 and 8. In addition to toxins, bacteria can also release enzymes that promote infection. The enzymes are not specific for any particular cell or cell receptor, but facilitate spread of the microorganism and are sometimes called invasins. A secondary effect may be considerable damage to the tissue and organs. The site of infection contains a broad spectrum of proteases, lipases, and deoxyribonucleases, and other enzymes secreted by microorganisms. The secretion of most toxic enzymes is common to broad groups of pathogens, whereas others discussed here are more limited to particular strains. Some proteases contribute to the defense against the immune and inflammatory system by digesting components of the plasma system, complement clotting, and fibrinolysis. Immunoglobulins, cytokines, and antimicrobial peptides. Most also weaken the surrounding connective tissue to allow invasion. Connective tissue is vulnerable to digestion by proteases that break down hyaluronic acid, collagen, fibrin, elastin, laminin, and glycosaminoglycans. Hyaluronidase is frequently produced by Streptococci, Staphylococci, and Clostridia to break down hyaluronic acid polymers in the matrix. Collagenase is a product of several Clostridiae, breaking down the intercellular matrix in the muscle during gangrene. Neuraminidase is especially useful for intestinal pathogens such as V. cholera and S. dysenteria, degrading the sialic acid that strengthens the intestinal epithelial junctions. Kinases such as streptokinase and staphylokinase activate plasminogen to digest large clots that interfere with bacterial spread. Many enzymes also will digest membranes of tissue cells, leading to lysis and release of nutrients that are useful to the microorganisms. Some hemolysins or leukocidins, so either hemolysins, those that lyse erythrocytes, or leukocidins, those that lyse phagocytes, are pore-forming toxins. But others are enzymes, lecithinases, or phospholipases that digest lipids in the membrane, resulting in hemolysis or cell death. C. perfringens produces a phospholipases and lecithinases that remove the polar head groups from cell membrane phospholipids or destroy phosphatidylcholine in the membrane of most cells. Whew, mouthful. Several enzymes perform a secondary role in invasion as adhesins to matrix components. Yersinia pestis, the plague, expresses a plasminogen activator protease that can activate plasminogen to plasmin, which can dissolve clots and activate collagenases. When expressed on the bacterial surface, it binds strongly to laminin in the matrix and facilitates invasion. As Agalactiae, a group B streptococcus, produces a serine protease, a C5a peptidase, that degrades a chemotactic factor C5a, but also has a high binding affinity for fibronectin. Destructive immune and inflammatory responses to bacterial infection. Much of the tissue damage caused by infections is secondary to inflammatory and immune responses. Infectious microorganisms routinely initiate an immune and inflammatory response, including infiltrates of phagocytic cells and a wide variety of T cells, production of antibody and activation of plasma systems via complement or clotting. Some are specific to antigens, T-cell antibodies expressed by the microorganisms, whereas others are not antigen-specific, phagocytic. Cytokines and chemokines secreted by the inflammatory and immune cells are designed to inhibit the proliferation and spread of infectious agents. However, most are also damaging to the surrounding tissue. Antibodies will react with antigens expressed by microorganisms. Overproduction of antigen released as soluble molecules will lead to reaction with antibody and formation of immune complexes in the circulation or in tissue and activation of inflammatory systems. Because the antigen is from an exogenous source, the mechanism of tissue destruction would be classified as allergic and would be type 2 if expressed on a cell surface, or type 3 if immune complexes are in the circulation. See Chapter 9. For example, individuals infected with S. pyogenes or T. pallidum are at risk for developing symptoms of type 3 hypersensitivity reactions, including kidney disease and vascular inflammation. The typical immune and inflammatory reaction against intracellular microorganisms or persistent infections involves a cell-mediated response with infiltrates of T cells and macrophages, as well as neutrophils. The inflammatory site is rich in pro-inflammatory cytokines, and as the individual's response increases, so do the destructive effects on surrounding healthy tissue. Many of these infections are very difficult to eradicate, leading to progressive increased production of cytokines. Persistent infections, like tuberculosis, may lead to formation of granulomas, which cause further destruction of the infected organ, such as the lung. The bacterial evasion of immune and inflammatory system. Evasion of the individual's immune and inflammatory system is multifaceted, and most successful pathogens incorporates several mechanisms. Rapid progression. Rapid progression is an evasive mechanism limited to bacteria and viruses because the primary immune response may take 3 to 5 days to reach protective levels. Some pathogens proliferate at rates that surpass the development of the immune system. An example is V. cholera, that causes severe vomiting and watery diarrhea, which has a 60% mortality rate and develops within 2 to 3 days of ingestion of the bacteria. Bacterial protection against phagocytosis and intracellular destruction. Anti-phagocytic capsules are expressed by most bacterial pathogens involved in pneumonia and meningitis. Capsules are mostly polysaccharide, although proteins also may be capsular components. Examples include the thick polysaccharide covering of the pneumococcus, the waxy capsule surrounding the tubercle bacillus, the polysaccharide slime capsule of P. aeruginosa, and the M protein of Pyogenes. Some bacterial toxins and extracellular enzymes kill phagocytic cells, such as the P. aeruginosa exotoxin A, staphylococcal hemolysin, and toxin from B. anthracis and pertussis decrease phagocytic and chemotactic activities and may be toxic to the cell. Streptococcal products, such as the streptolysin O, bind to cholesterol in the membrane of the phagosome and initiate destruction through the cytoplasmic release of lysosomal enzymes. Normally, a macrophage would effectively kill bacteria, but mechanisms for intracellular survival have evolved to benefit of the infectious agent. Intracellular bacteria can survive and even multiply in macrophages, the Brucella, Listeria, leprosy 1, tuberculosis, or in other cells. Bacterial killing results from fusion of lysosomal granules with the phagosome to produce a phagolysosome and activation of the oxygen-dependent and oxygen-independent mechanisms. Tuberculosis produces toxins that prevent phagosomal-lysosome fusion so that the environment in the phagosome remain relatively non-toxic. A second means of avoiding killing is neutralization of the toxic environment in the phagolysosome. Production of catalase and superoxide dismutase, such as in gonorrhea and Brucella abortus, destroys toxic oxygen products produced by the hexose monophosphate shunt. And then there is a third means of avoiding killing, is escape from the phagosome. Lysins, hemolysins of Shigella, Listeriolysin O, and phospholipase C in L-monocytogenes, and phospholipids A and rickets are secreted enzymes that break down the phagosome membrane, releasing the bacteria into the cytoplasm where they multiply. By blocking recognition of bacterial antigens, pathogens that coat themselves with human proteins may fool the immune system by masking antigens. Some bacteria surface proteins, protein G and S. Pyogenes, bind to the Fc portion of antibody, thus forming a protective coat of self-protein. Binding through the Fc holds the antibody in an orientation that does not allow complement activation or phagocytosis. T. pallidum coats itself with fibronectin capsules, may contain sialic acid, such as E. coli K12, which closely resembles the sialic acid on the surface of most human cells, or the uronic acid, which is the basic substance in normal connective tissue. The antigenic variation allows the pathogen to alter surface antigens that are targets of protective immune responses. Thus, as the individual develops protective levels of antibodies, the pathogen responds by changing antigens and becoming resistant. The three primary mechanisms of antigenic variation are mutation, a change error in the gene structure that results in a variant expression of antigen; recombination, the exchange of DNA fragments between cells resulting in a new DNA sequence and variants of antigen expression; and gene switching, the switching of genes on and off resulting in variation of antigen expression. Neisseria uses pili to adhere to epithelium. An antibody against these antigens can abrogate adherence. Neisseria undergoes phase shifts during which pillar antigens are changed by progressively silencing one of one set of 10 or 11 available pili genes, and activation of the others that express antigenic types or serotypes across the species. At least 80 different strains of group A streptococci express different serotypes of the capsular M protein. S. pneumoniae has at least 100 different serotypes based on capsular polysaccharides. Some gram-negative bacteria can revert to Ref forms, in which serotype-specific carbohydrates are deleted, thus becoming resistant to antibody and activation of complement. Blocking the immune response to bacteria, bacteria may spontaneously release surface molecules that bind to and neutralize antibody. These include endotoxins from gram-negative bacteria and capsular antigens from S. pneumoniae and N. meningitis. The secretion of large amounts of soluble antigen may lead to formation of immune complexes. Bacteria may break down molecules of the immune or inflammatory system. An IgA protease produced by meningitis causes microorganisms and other related bacteria. In gonorrhea and meningitis, H. influenza, S. pneumonia cleaves IgA at the hinge region into ineffective Fc and Fab regions. Salmonella membranes, proteases degrades nonspecific antimicrobial molecules like defensins and cathelicidins. Complement is a major component of the defense against bacterial infections through production of opsonin, or C3b, and chemotactic factors, C3a, C5a, for neutrophils. Teichoic acid in the gram-positive cell wall provides resistance against complement-mediated lysis. Bacterial regulatory proteins, Borrelia complement regulatory acquired protein, Neisseria porins, and members of the Streptococcus M protein family affect complement activation, including destabilization of the C3 convertase complex or degradation of the opsonin C3b. Pseudomonas produces elastase, which breaks down C3 of the complement system, and the 5,6-KDA protease breaks down the chemotactic factor C5a. Some bacterial pathogens can broadly suppress immune responses against their own antigens, as well as other antigens unrelated to the infectious agent. Chronic bacterial infections like leprosy and tuberculosis induce anergy, suppressed response to multiple antigens, in infected hosts. The Heliobacter pylori, or H. pylori, releases LPS that binds to the dendritic cells and blocks development of T helper 1 cells, produces toxins that block the T cell interleukin-2 receptor signaling pathway, thus inhibiting maturation of Th cells, and induces the Th17 response that impairs inflammatory responses. One of the most common bacterial infections is the Staphylococcus aureus. All right. So that was bacteria. Jumping into infectious fungi. Fungi are eukaryotic microorganisms with thick, rigid cell walls and the capacity to form a variety of complex structures. Fungi may grow as a mold with branched filaments or as a meshwork mycelium structure, such as aspergillus or causing aspergillosis. Yeast with ovoid or spherical shapes, Candida albicans, which causes candidiasis, or dimorphic, with a yeast-like appearance in tissue, and mycelium in culture. Histoplasma capsulatum, which causes histoplasmosis, a respiratory disease. Molds are aerobic, and yeasts are facultative anaerobics. The cell wall is composed of polysaccharides that differ from the peptidoglycans of bacteria and are resistant to bacterial cell wall inhibitors, such as penicillin and cephalosporins. In contrast to bacteria, the cytosol of fungi contains organelles, mitochondria, Golgi apparatus, microtubules, microvesicles, endoplasmic reticulum, and nuclei. Many of the antifungal drugs, amphotericin B or ketoconazole or fluconazole, used to treat deep or systemic infections are toxic to the host because the fungal cell composition is similar to that of the human cell. Common pathologic fungi are summarized in Table 10.4. So common fungi are dermatophytes, Trichophyton, Candida albicans, Sporothrix, Stachybotrys or black mold, Coccidioides, Histoplasma capsulatum, Aspergillus, Pneumocystis, Cryptococcus neoformans, Candida albicans. All right. Infections or infection with a fungus is called a mycosis. Many pathogenic fungi, H. capsulatum, Coccidioides immitis, or Blastomyces dermatitis, grow as saprophytes in the environment and are transmitted by inhalation or contamination of wounds. Other medically relevant fungi either exist as human commensal or cause relatively mild infections. Superficial mycosis of the skin, nail, hair, and mucous membranes of the mouth and vagina. These include dermatophytes and yeast. Human-to-human transmission is the primary means of transmission of dermatophytes. For instance, tinea pedis, athlete's foot, T. capititis, ringworm or scalp, and T. unguineum, fungal infection of nails. Systemic mycosis generally result from inhalation of spores present in the contaminated environment and initially present as a pulmonary infection. Infection that disseminates to other organs can be life-threatening. Systemic mycosis caused by the opportunistic fungi is usually secondary to immunosuppression caused by genetic defects. Infections such as HIV, cancer, and drugs used to prevent transplant rejection. Fungi are diagnosed by microscopic observation of specimens treated with potassium hydroxide and stained to enhance visualization of spheres and filaments. Specimens also can be cultured. Skin tests are available for species of Aspergillus. The mechanism of fungal invasion and tissue destruction. Specific adherence to the epithelium is provided by several polysaccharides on the fungal surface. Glucan, mannan, glycoprotein, and chitin molecules adhere with host receptors, including toll-like receptors, mannose receptors, and cadherins. A cell wall adhesion molecule, agglutinin-like sequence 3, on several fungi promotes adherence to epithelial cells as well as silicone, thus facilitating infection of implants and other medical devices. Pneumocystitis jirovecii infection is a major concern in the individual with AIDS. As with many fungi, pneumocystitis has two life cycles, a cyst form and a trophic form. The cyst form is dormant, more globular, with a thick wall, and when ruptured releases multiple spores. The trophic form, or trophozoite, is the infectious and proliferative form. Two surface proteins, glycoprotein A and major surface glycoprotein, mediate attachment of the trophozoite to alveolar epithelial cells, leading to the development of pneumonia. Pathogenic fungi. Fungal toxins and products. Pathogenic fungi do not commonly produce toxins. As virulence factors, the only identified peptide toxin related to virulence is a cytolytic toxin, the Candida lysine, that is crucial for C. albicans infection of mucosa. A sulfur-containing mycotoxin that induces apoptosis of many cells of the inflammatory and immune system is produced by A. fumigatus. Several toxins secreted by mold in the environment cause disease without fungal infection. Mycotoxins are produced by molds that grown on nuts, beans, and grains. Ingestion of these toxins affect muscle coordination, causes tremor, and may be fatal. Other fungal toxins may cause cancer. Aflatoxins produced by some Aspergillus are especially carcinogenic. By mechanism similar to those described for bacteria, fungal infection damage tissue directly by secretion of enzymes and indirectly by initiation of an inflammatory response. Secreted enzymes such as proteases, phospholipidases, elastases, damaging cells, and intercellular matrix, leading to breakdown of tissue and cellular necrosis. Destructive immune and inflammatory responses to fungal infection. A typical immune and inflammatory reaction against fungal infections involve a cell-mediated response with infiltration of T cells, macrophages, and neutrophils. Fungal infections can be very difficult for these systems to eradicate, leading to progressively increasing production of pro-inflammatory cytokines. Hyperinflammatory responses enhance virulence of A. fumigatus by increasing adhesion and invasiveness. Thus, the host's response increases, so do the destructive events on surrounding healthy tissue. In cases of persistent infection, granulomas form and compromise the normal function of the infected tissue. Pulmonary exposure to spores and soluble antigens also may elicit an IgE response and allergic pneumonitis, the inflammation of the lungs. Examples include allergic bronchopulmonary aspergillus and allergic fungal sinusitis, both of which are type 1 allergic reactions. Excessive degranulation of mast cells may even cause anaphylactic shock. The host defense against fungal infection include the fungistatic properties of neutrophils and macrophages. T lymphocytes are crucial in limiting the extent of infection and producing cytokines to further activate macrophages. Resistance to the individual's protective mechanism is a complex process that requires the expression of multiple genes at different stages and different sites of infection. Pathologic fungi are generally dimorphic and readily adapt to the host environment, responding to temperature variations, low oxygen levels, more alkaline pH, and other conditions in the host tissue by undergoing changes in morphology and switching from avirulent mold forms to virulent yeast forms. Similar conditions also trigger yeast, like C. albicans, to switch from the yeast form to its more virulent hyphal form. As with parasites, rapid progression is not a major factor in immune evasion by fungi. The fungal protection against phagocytosis and intracellular destruction. Encapsulated yeast, Cryptococcus neoformans, are more resistant to the phagocytosis than unencapsulated yeast. A capsule is antiphagocytic by masking surface PAMPs, pathogen-associated molecule patterns, and inhibiting recognition by PRRs, the pattern recognition receptors, on phagocytes and epithelial cells. Aspergillus fumigatus and many other fungi produce toxic metabolites that inhibit macrophage and neutrophil phagocytosis. Molecules like gliotoxin may suppress inflammation, including suppression of mast cells, activation, degranulation, and secretion of leukotrienes and cytokines. Even if successfully ingested by phagocytic cells, fungi have developed complex processes for intracellular survival. Pathologic fungi are generally dimorphic and readily adapt to the host's environment, responding to temperature variation, low oxygen level, more alkaline pH, and other conditions in the host's tissue to undergoing changes in morphology and switching from avirulent mold forms to virulent yeast forms. The yeast form of histoplasma replicates in phagosomes and phagolysosomes. Some yeasts also produce proteins that inhibit the activity of lysosomal proteases. The macrophage may be destroyed as a result of membrane modification by the fungus, or in the case of histoplasma, may harbor the fungus in granulomas. Breakdown in the granulomas over time result in release of viable fungi and recurrence of the infection. Blocking recognition of fungal antigens. Altered antigen expression affords protection against the developing immune response. Although this defense strategy is used only by selected fungal pathogens, pneumocystitis contains approximately 80 different GPA and MSG genes, and only one of which is expressed at a time. Modulation of these antigens provides resistance against immune destruction. The blocking the immune response, several yeasts stimulate the production of immunosuppressive cytokines, resulting in downregulation of the individual's immune response. The yeast C. neoformans suppresses inflammation by inhibiting production of the pro-inflammatory cytokines TNF-A, or tumor necrosing factor alpha, and interleukin 12, and inducing production of the anti-inflammatory cytokine interleukin 10. The overall result is suppression of macrophage function and protection against killing. Candida albicans is a common cause of infection, which is our box to read here. So that leads us to our infectious parasites and protozoans, which we are skipping right on over. We're going to jump to page 10,007. Where we got like another, I don't know, let's say 30 pages to go. So, jumping straight into HIV and the AIDS acquired immunodeficiency syndrome. So, HIV and AIDS. The most notable form of secondary or acquired immunodeficiency is caused by the HIV virus. HIV causes AIDS. HIV is a retrovirus that infects and depletes a portion of the immune system, CD4 plus Th cells, making individuals extremely susceptible to life-threatening infections and malignancies. When AIDS was recognized as a major health epidemic in sub-Saharan Africa and in the US in the 1980s, an AIDS diagnosis was considered a death sentence with near 100% mortality rate within 2 years of diagnosis of AIDS. With the advent of aggressive treatment with antiretroviral drugs in the mid-1990s, the widespread public health campaigns HIV and AIDS has become a chronic and manageable illness, with many infected individuals having a near normal lifespan. Currently, about half of the 18 million individuals living with HIV/AIDS in Africa are being administered antiretroviral drugs. As yet, however, antiviral medications are not fully available at an affordable price in many parts of the world where HIV and AIDS remain the major cause of morbidity and mortality. The transmission of HIV in Africa is predominantly heterosexual, where the distribution of individuals with HIV and AIDS in the United States reflects the primary means of transmission within the original cohort. Although isolated cases of what was later identified as AIDS occurred in the United States as early as 1959, the appearance of the AIDS epidemic has been linked to several gay males who had contracted HIV infection during visits to the Caribbean and probably Haiti. I'm not sure why that needs to be said. Initial reports of outbreak in the United States were of gay men who developed pneumocystitis carinii pneumonia and aggressive Kaposi sarcoma, both of which were uncommon and mostly observed in immune-deficient individuals. The epidemic then spread to intravenous drug abusers and heterosexuals. In 2014, the latest available statistic, 44,073 men and women were newly diagnosed with HIV infection, with gay or bisexual men representing 67% of all individuals and 83% newly diagnosed men. Women represented 19% of the newly diagnosed individuals, with 87% of women contracting HIV through heterosexual encounters. As a result of aggressive antiretroviral therapy and public education, the transmission to women had declined by about 40% over the previous decade. About 6% of all individuals newly diagnosed with HIV were associated with IV drug abuse. In 2015, 18,000 people were diagnosed with AIDS, and since the early 1980s, 1,216,000 people have been diagnosed with AIDS. In 2014, 6,700 deaths were attributed directly to HIV infection. An estimated 650,000 individuals with a diagnosis of AIDS have died since the early 1980s. The transmission of HIV is a blood-borne pathogen that presents in body fluids with the typical routes of transmission: blood or blood products, IV drug abuse, heterosexual and homosexual activity, and maternal child transmission during childbirth. The CDC estimates that 91% of new HIV infections in the US result from sexual activity with those who are undiagnosed or diagnosed but not treated for HIV. As with all blood-borne infections, healthcare providers are at increased risk of contracting the infection from the individual's blood, since the strict implementation of universal precaution and the use of post-exposure prophylaxis with antiretroviral drugs, infection of healthcare providers is extremely rare. Thank the good Lord. HIV-1 was initially isolated by researchers at the Pasteur Institute as the lymphadenopathy or AIDS virus, a discovery for which they received a 2008 Nobel Prize in Medicine. A second major and less virulent variant, HIV-2, was identified later and is found mostly in Western Africa. HIV is a member of the retrovirus family, which carries genetic information in the form of two identical copies of single-stranded RNA. The RNA copies are packaged in the viral particle within a capsid constructed of viral capsid protein P24. The capsid also contains two viral enzymes, reverse transcriptase and integrase. Retroviral particles are contained in a plasma membrane envelope originating during release from the infected cell. The membrane contains two viral proteins, GP41 and GP120, referred to as envelope proteins. Between the envelope capsid are viral matrix proteins underlying the environment or envelope and another viral enzyme, a protease. As with all other viruses, the life cycle must begin with attachment and entrance into the cellular target. HIV is primarily tropic for CD4 T helper cells. Although macrophages and dendritic cells also express CD4 and can be infected, attachment occurs between the viral envelopment proteins and CD4. The envelope protein GP41 is anchored in the plasma membrane and is referred to as TM or transmembrane protein. The N-terminal portion of the TM consists of very hydrophobic amino acids. The TM associates with a GP120 envelope protein called the SU or surface protein, which contains binding sites for CD4. When the surface protein attaches to CD4, it undergoes a conformational change that allows further recognition of the cell surface chemokine receptors, primarily CCR5, and exposure of the TM hydrophobic tail, which interacts with the lipid region of the T cell and initiates a process of fusion between the viral envelope and the cellular membrane. The fusion event releases a viral capsid into the cellular cytoplasm, where it undergoes undercoating, shedding of capsid proteins, and release of viral RNA enzymes. The third step in the life cycle, integration, is controlled by reverse transcriptase, which converts single-stranded viral RNA into double-stranded DNA. The viral integrase inserts viral DNA into the infected cell's DNA, where it may remain dormant. If replication is activated, the GAG gene encodes a large precursor protein that undergoes post-translational modification by the viral protease to generate smaller matrix, so our P17, and capsid proteins, P24. The ENV gene encodes a large GP160 glycoprotein that is processed by a cellular protease furon into the TM and SU proteins. The capsid and nuclear component of the virus undergo assembly in the cytoplasm, and the ENV proteins are inserted into the plasma membrane. The assembled particles buds from the cell surface as the infectious virion. The major immunologic findings in AIDS is the striking decrease in the number of CD4 Th cells. Several mechanisms of Th cell killing are used. The replication and release of new virion triggers apoptotic cell death of the infected cell. The release of death ligands, the Fas ligand and TNF, initiates apoptosis in neighboring uninfected Th cells. The infected Th cells also becomes a target for killing by the CD8 T cytotoxic cells. So what kind of clinical manifestations do we have with HIV? With the advent of antiretroviral drugs, the early identification of HIV infection is essential. The CDC has issued a new system for staging HIV infection at diagnosis and during monitoring and therapeutic effectiveness and managing the disease. The stages are adapted for every age group and based primarily on diagnostic laboratory tests. Stage 0 disease covers the first 180 days after infection, and stages 1 to 3 are based primarily on the CD4 cell count to monitor disease progression. The lymphoid areas of the mucosal surface are the primary sites of initial infection, particularly if the mucosa has been damaged. Dendritic cells and mucosal T cells probably spread the infection to other peripheral lymphoid organs, especially follicular dendritic cells in the lymph nodes. Production of new viruses, particles, proceeds rapidly with increasing viremia. HIV RNA may be detected in the plasma by 10 days after an acute infection, and HIV P24 antigen detection about 4 to 10 days after viral RNA. Indication of an individual's immune response quickly follows. HIV-specific IgM antibody is detected about 3 to 5 days after P24. Antigen and levels of IgG begin rising soon afterwards and remain elevated throughout the disease process. As with most immune responses against viral diseases, the IgM levels are usually transient. The appearance of viral RNA in the plasma is indicative of viremia and the appearance of infectious virus in the body fluids, an indication that infected individuals can transmit HIV to other individuals. 40 to 90% of individuals in stage 0 will experience symptoms of acute viral syndrome. Symptoms may include fever, fatigue, headache, lymphadenopathy, pharyngitis, myalgia or muscle pain, arthralgia, joint pain, and a skin rash that can be extensive. Without knowledge of exposure to HIV, the infected individual may suspect a self-limiting influenza or infectious mononucleosis condition. By 180 days, the initial burst of viremia and acute viral syndrome will have resolved. The infected individual will be asymptomatic, yet infectious HIV is present in the body fluids, and the process of HIV-mediated destruction of CD4 cells has just begun. Stages 1 through 3 are determined by diminishing CD4 T cell counts, adjusted for normal age-related levels. In the first year of life, the normal CD4 T cell count is the highest and decreases with age. For instance, stage 1 disease is classified after 180 days of infection, and the CD4 T cell counts are still normal, greater than 150,000 cells. Then after 1 year, it's about greater than 1,000. At 1 through 5 years, it's greater than 500. At 6 years to adult, stage 2 is characterized by the diminished number of CD T cells, 750 to 1499. At greater than 1 year, 500 to 900. So it's a diminishing number. At stage 3 is defined as AIDS and is determined by the CD4 cell counts of less than 750, 500, or 200 cells per UL for the respective age groups of the development of AIDS. Then there is a list of AIDS-defining opportunistic infections and neoplasms found in individuals with AIDS. The treatment and prevention of HIV. The current regimen of highly active antiretroviral therapy, or HART, for treatment of HIV infection is a combination of drugs that attack different portions of the viral replication pathway. Since the FDA approved of zidovudine (AZT), more than 25 antiretroviral agents representing six different classes have been approved to treat HIV and AIDS, and several others are in the various stages of testing. Classes of antiretroviral agents include inhibitors of the enzyme reverse transcriptase, nucleoside-nucleotide reverse transcriptase inhibitors, NRTIs, and non-nucleoside reversal transcriptase inhibitors. Inhibitors of the viral protease entry and intercellular fusion inhibitors that prevent attachment of virus to the target cell or prevent fusion between HIV and the cell membrane. And inhibitors of the viral integrase enzyme. Integrase strand transfer inhibitor INSTIs. A sixth group of drugs are pharmacokinetic enhancers, such as cobicistat or COBI, that are administered with some P and INSTIs. Enhancers affect intestinal transport proteins and liver enzymes that would normally break down some antiretroviral drugs, thus allowing smaller doses to be used. Many other drugs are in clinical testing, including GAG maturation inhibitors that prevent enzymatic activation of the GAG precursor protein, and GP120 inhibitors prevent conformational changes necessary for attachment and entry process, and broadly neutralizing antibodies bind to proteins on viral envelope and prevent attachment. The recommended first-line treatment is a combination of two NRTIs and a third drug from another class. Many drug combinations are available in a single pill, facilitating compliance. The specific regimen and drugs may vary based on a variety of an individual's characteristic. Other drugs, like entry inhibitors, are reserved for those who develop resistance to first-line drugs. The goals of therapy are reduction of viral load in fluids, reduced HIV-associated morbidity, prolonged survival, and prevention of HIV transmission. The plasma viral load is frequently suppressed below detectable levels within weeks. With treatment beginning at the time of diagnosis, transmission has decreased about 90%. Retroviral preventative treatment of uninfected partners of those who are HIV infected has diminished transmission by 96%. Without treatment, the progression from acute infection to AIDS takes 9 to 10 years, with deaths occurring within 2 to 3 years. Death from AIDS-related diseases has been reduced significantly since the introduction of HART. Those who respond well to HART may survive for several decades. Continued suppression of HIV and AIDS progression absolutely requires daily and continuous use of medication. However, many people do not respond to HART therapy. Those who do not respond are not cured, and resistant variants to these drugs have been identified. Individuals who initiate antiretroviral therapy will still have a shortened life expectancy secondary to HIV-associated persistent immune activation. Continued systemic inflammation secondary to persistent HIV infection and toxicity of available drugs increase the risk for developing chronic and potentially fatal morbidities. Complications include dyslipidemia, imbalance of blood lipids, insulin resistance, lipodystrophy, changes in body composition, either loss or accumulation of fat, decreased bone density, and increase in incidence of fractures, diabetes, renal disease, and liver disease. The risk of cardiovascular disease is increased twofold. Antiretroviral drug therapy does not cure HIV or AIDS because the HIV genome incorporates into the DNA of the infected cells that may act as reservoirs and release viral particles if therapy is stopped. The primary reservoir is probably long-lived CD4 memory T cells, although contribution to the reservoir may include the infected cells, such as macrophages, dendritic cells, astrocytes, microglia, and hematopoietic progenitors. HIV also may persist in regions where the antiviral drugs are not as effective, such as the central nervous system. HIV penetrates the central nervous system early in the infection and persists despite successful treatment with antiretroviral drugs. Neural infection leads to viral encephalopathies and apoptosis of neurons. The incidence of HIV-associated dementia was about 20% in untreated individuals, but antiretroviral therapy has reduced that number to about 5% and decreased the severity. Dementia is now rarely seen. Ultimately, the best hope for preventing the spread of HIV infection is development of an effective vaccine. Most common antiviral vaccines, rubella, mumps, influenza, induce protective antibodies that block the initial infection. Only one vaccine, rabies, is used after the infection has occurred. That approach is successful because the rabies virus proliferates and spreads very slowly. Whether an HIV vaccine would be effective is either preventing or treating HIV infection is problematic. And the results of recent vaccine trials have, for the moment, dampened enthusiasm. Characteristics of HIV have been difficult to overcome. HIV-1 commonly undergoes random mutation that result in an extreme and changing antigenic diversity. During transmission, bodily fluids may contain a variety of viral antigenic types, so that effective vaccines may need to induce protection against a broad spectrum of HIV surface antigens. Pediatric HIV. Neonates become HIV infected from their mothers by the passage of the virus across the placenta during pregnancy. By contact with infected maternal blood at childbirth or through the milk during breastfeeding, the transmission rate in the United States was about 13%, but has been reduced to 1% with the use of prenatal antiviral therapy beginning early in pregnancy, the implementation of neonatal prophylaxis, and the avoidance of breastfeeding. Without prenatal treatment, symptoms of HIV infection usually develops within 6 months of neonatal life. 20% get opportunistic infections within the first year, and life expectancy is generally less than 3 years. Because of aggressive clinical intervention, there are currently fewer than 50 cases of pediatric HIV infection diagnosed yearly in the United States. The presence of passive maternal antibody limits the use of HIV antibody testing in infants in the high-risk category up to 18 months of age. The current recommended diagnostic procedure is testing initially for HIV nucleic acid, viral RNA in plasma, or DNA within cells. Determination of whether the child is infected is based on repeated negative test results. 2 negative nucleic acid test results at 2 and 4 weeks after birth is considered a presumptive uninfected state. A definitive uninfective is defined as 2 nucleic acid tests results at 1 and 4 months. Combined heart is initiated immediately in infants and leads to a reduced viral latency of 76% reduction in AIDS-related mortality, and many children reaching the adolescent period with functional immune systems. Complications in children undergoing heart are related to metabolic disorders, including insulin resistance and dyslipidemia. Because of the metabolic complications of heart and the ongoing HIV-associated inflammation, half of HIV-infected children are at risk of coronary disease. Most HIV-infected children who reach adulthood will have indication of coronary artery abnormalities. A third or less of children will present with low bone density, but with no increase in fractures. A particular vulnerable site of infection in infants and children is their CNS, or central nervous system. HIV encephalopathy is more common in the advanced stages because survival of children with HIV has been prolonged with effective treatment. The incidence of progressive encephalopathy has increased. For diagnosis of HIV-related neurologic effects, at least one of the following progressive findings needs to be present for at least 2 months. In the absence of the concurrent illness other than HIV that could explain the following findings: failure to attain or loss of developmental milestones or loss of intellectual ability, verified by a standard developmental scale or neuropsychologic test. Impaired brain growth or acquired microcephaly demonstrated by head circumference measurements or brain atrophy demonstrated by computated or CT scans or MRIs, with serial imaging required in children less than 2 years of age. Acquired symmetrical motor deficits manifested by affecting a child 1 month of age or older. The onset of progressive encephalopathy may be a prognostic indicator of a poor outcome. Other insults may accompany HIV in a young child and affect growth and development, such as drug exposure, prematurity, chronic illness, and a chaotic social atmosphere. The pathogenesis of HIV encephalopathy in children is poorly understood, but the presence of inflammatory mediators may be a contributing factor. So, countermeasures against pathogens. An extremely effective means of countering infectious microorganisms is rigorous use of environmental infection control measures, including control of insect vector populations, establishment of modern sanitation facilities, provision of clean water and uncontaminated food supplies, and other measures. Additionally, prophylactic and therapeutic procedures have been developed to prevent pathogens from initiating diseases, such as vaccines, or to subdue the pathogen once the disease process has started, antimicrobials. Vaccine development has focused successfully on preventing the most severe and common infections. With the initial success of antibiotic therapy, there was no perceived need for vaccination against many common and non-life-threatening infections. The increasing problem of antibiotic-resistant pathogens, however, forces a reappraisal of that strategy, and a greater emphasis is being placed on the development of new vaccines. Infection control measures. Although effective means of safeguarding population from exposure to infectious diseases are well known, lack of implementation or breakdown in application of these initiatives have led to the re-emergence of some infectious diseases. These diseases include cholera, dengue fever, and the West Nile virus. Sewage removal and other public health initiatives are now the expected norm in developed countries. The quality of water and food, as well as disposal of human and animal waste, remains poor in many developing countries. Rapid urbanization, as well as other problems, poverty, overcrowding, mass relocation of people because of war, rapid destruction of forests, has put pressure on already inadequate systems. In 1991, outbreak of cholera in Peru resulted in an estimated 1,100 deaths in Peru and more than 4,000 deaths in Latin America, and was attributed to problems with infrastructure, including inadequate sanitation. Additionally, previously successful programs designed to control the breeding of insect vectors have been reversed. Despite an international emphasis on draining standard water that provides breeding grounds for mosquitoes, large unmanaged areas still abound. A very successful international mosquito eradication program resulted in decreasing incidence of mosquito-borne diseases. Some regions were declared free of mosquito-borne diseases. However, lack of access to pesticides and failure or delay in initiating fumigation and larvicide programs for mosquitoes has resulted in rebound of mosquito-related diseases. In previously disease-free regions of some parts of the world, the consequence is increasingly severe outbreaks of viral disease. An example is outbreaks of dengue virus and West Nile virus in areas of India in the last several years. This problem is exacerbated by the development of insecticide-resistant mosquitoes. Antimicrobials are natural products of fungi, bacteria, and related microorganisms and kill or inhibit the growth of other microorganisms. They are basically microbial defense mechanisms by which one microorganism can defend itself against another. The goal of antibiotic therapy is elimination of the pathogenic microorganism. Some antibacterial antibiotics are bactericidal, whereas others are bacteriostatic, which inhibit growth until the organism is destroyed. The mechanism of action of most antibiotics are inhibition of the function or production of the cell wall, prevention of protein synthesis and blockage of DNA replication, or interference with folic acid metabolism. Because viruses use a biochemical pathway of the host's cells, they have been far less success in developing specific antiviral antibiotics. Antibiotic resistance observed clinically is of a major importance and has become a problem worldwide. As the use of penicillin became widespread, Alexander Fleming, the discoverer of penicillin, frequently warned about the development of penicillin resistance, and the first bacterial penicillinase enzyme that destroyed penicillin, B-lactamase, was prescribed in 1940. At least 1,000 variations of B-lactamase have since been discovered, being natural products of microorganisms that exist in nature. Resistance mechanisms also develop in nature. Genes related to antibiotic resistance have been characterized from samples of permafrost that were dated at 30,000 years ago from the oral microbiome of 2,000-year-old skeletons, from the gut microbiome of a Peruvian mummy dated nearly 1,000 years after death, or AD, from several other samples that greatly predate the era of therapeutic antibiotics. As might be expected, environment antibiotic resistance has increased greatly since the use of antibiotics in agriculture. The CDC publishes a list of antibiotic-resistant threats ranked as urgent, serious, and concerning at the following websites. They estimate that more than 2 million antibiotic-resistant illnesses occur in the US yearly, resulting in nearly 23,000 deaths. The trend is progressing upward, more infectious agents being resistant to multiple antibiotics, such as the classification of extensively drug-resistant TB that is resistant to all of the first and second-line drugs, and the cases of some vancomycin-resistant infections that are resistant to almost all antibiotics. Antibiotic resistance may result from a variety of inherited processes that coexist in different combinations in the same microorganism. Resistance genes can be spread within the bacterial community by horizontal gene transfer. Genetic information can be transferred by means of transformation, uptake of short DNA fragments from the environment, by transduction, transfer of DNA by means of viruses that infect bacteria, bacteriophages, or by conjugation. Transfer of DNA, usually in the form of circular plasmids through pili connecting the bacteria. A single plasmid may contain a large amount of genetic information from multiple types of antibiotic resistance. Enzymatic inactivation of antibiotics is one of the most common forms of resistance. The B-lactam antibiotics contain a B-lactam ring central to their structure and are bactericidal by inhibiting the formation of the bacterial cell wall. The initial class of B-lactam antibiotics, penicillin cephalosporins, blocked synthesis of gram-positive cell wall, where newer broad-spectrum B-lactam antibiotics, carboxypenicillins, also affect gram-negative cell wall synthesis. Resistance is conferred by enzymatic B-lactamase, destruction of the antibiotic. Another class of antibiotics, aminoglycosides, streptomycin, neomycin, gentamicin, is bactericidal primarily to gram-negative aerobic bacteria and against tuberculosis by binding to sites on ribosomal RNA and inhibiting protein synthesis. Resistance is conferred to aminoglycoside modifying enzymes, which acetylate and inactivate aminoglycoside. Another major form of antibiotic resistance is caused by multi-drug resistance transporters. MDRs are members of the diverse and widely expressed family of transmembrane proteins that are designated or designed to protect the cell by diminishing the rate of intracellular accumulation of antimicrobials, by preventing entrance or, more commonly, increasing active efflux of the antibiotic. At least 6 different bacterial MDR families have been identified, with their genes also primarily carried on plasmids that can be transferred to other bacteria. Expression of MDRs is not limited to bacteria. C. albicans uses multiple MDRs as efflux pumps for antibiotics. Individual MDRs are capable of transporting and simultaneously protecting microorganisms from several antibiotics. Kanamycin, erythromycin, doxycycline. Other methods of antibiotic resistance are less common, but still important in understanding the combination of protective mechanism leading to multi-drug resistance. Antibiotics gain entrance to bacteria by passing through the cell wall. Resistance may result from sequestration or decreased uptake of the antibiotic. Thickened cell walls or biofilms trap antibiotics into the extracellular matrix to prevent access to bacteria. Decreased uptake also may result from modification of the cell wall. P. aeruginosa undergoes minor changes in cell wall chemistry and becomes resistant to the uptake of B-lactam antibiotics. Additional mechanisms protect the molecular target of particular antibiotics. The target may be altered by mutation or other means. Genetic amplification and overproduction of the target molecule result in excessive numbers of targets and reduced antibiotic sensitivity, increased resistance to sulfonamides in some streptococci. Target bypass is a resistance mechanism resulting from switch from an antibiotic-sensitive metabolic to one that is more antibiotic-resistant. Some bacteria have gained resistance to sulfonamide by converting from use of environmental para-aminobenzoic acid for conversion to folic acid, which is an antibiotic-sensitive step in nucleic acid synthesis, to the direct use of environmental folic acid. The target molecule also can be protected by novel bacterial proteins that block the antibiotic binding site. Multiple antibiotic resistance results from the simultaneous application of a combination of mechanisms. Methicillin-resistance, or methicillin-resistant Staphylococcus aureus, or MRSA, caused an international healthcare crisis. Outside of the United States, MRSA accounts for 80% of S. aureus infection. MRSA produces a B-lactamase, normally B-lactamase-resistant broad-spectrum antibiotics, methicillin, can be substituted. However, MRSA carries an extra chromosome gene, MECA, for resistance to methicillin. The MECA gene encodes a penicillin binding protein with a lower affinity for B-lactam and therefore less sensitivity to inhibition by antibiotics. Resistance to aminoglycoside antibiotics is accomplished by an expression of AMEs, production of excess para-aminobenzoic acid, and spontaneous mutation of RNA with a lower affinity for antibiotics. Resistance to the glycopeptide antibiotic vancomycin is controlled by a gene VAN-A, which result in alterations in peptidoglycans and loss of vancomycin binding site. Bless you. Antibiotic sequestration also occurs in the thick peptidoglycan layer and within biofilms. MRSA expresses 4 different classes of multi-drug-resistant transporters. Fusidic acid, one of the few drugs for MRSA, binds to the ribosome and prevents protein synthesis. Resistance results from a small protein that binds to the target and prevents interactions with the antibiotic. Multiple antibiotic resistance has appeared and can be related to overuse of antibiotics and not completing an antibiotic regimen. Overuse of antibiotics leads to the destruction of the normal microbiome, allowing the selective overgrowth of antibiotic-resistant stains or pathogens that have previously been kept under control. For example, treatment with clindamycin can compromise the normal intestinal microbiome, allowing the overgrowth of C. difficile and the development of pseudomembranous colitis. Lack of compliance in completing a therapeutic antibiotic regimen allows the selective resurgence of microorganisms that are more relatively resistant to the antibiotic. With the development of multiple antibiotic-resistant strains, creativity in addressing this challenge must be rekindled. New antibiotics may solve a portion of the problem, or may exacerbate it further as pathogens develop resistance to new antibiotics as well. Available antibiotics that prove to be no longer effective must be replaced with alternative forms of therapy, including the increased development and use of vaccines. In that context, an old treatment has been revisited, the admiration of the lytic bacteriophages. Bacteriophages are viruses that are specifically infect bacteria, and in some cases of lytic phages, result in bacterial death and lysis. The advantages of bacteriophage therapy are numerous. Cocktails of complementary phages can be used. Phages can progressively infiltrate a biofilm that trap antibiotics, and phages can be genetically engineered to express enzymes that weaken the biofilm. Bacterial resistance to phages may occur related to alteration of phage receptors on the bacteria. However, this therapeutic agent clearly needs to be explored further, and studies in progress to test the effect of bacteriophage therapy on P. aeruginosa chronic lung infections. Active immunizations and vaccines. Contracting and surviving an infectious disease is the most effective means of developing lifelong immunity against a particular pathogen. However, some infections cause a great deal of morbidity and mortality. The purpose of vaccination is to induce active immunologic protection before exposure to the risk of debilitating or fatal infections. For each vaccine, an initial immunization protocol is developed to produce large numbers of memory cells and a sustained protective secondary immune response in the greatest number of individuals. Even under optimized protocols, vaccines are not complement or completely protective. For every vaccine, a small percentage of recipients do not produce a protective immunity response, referred to as primary vaccine failure. Failure to induce an immune response occurs in about 10% of recipients of measles or rubella, mumps, rubella, and hepatitis B vaccines. And an activated influenza vaccine may only protect 30% to 40% of the 65 years or older population. A recent approved high-dose injectable vaccine with 4 times the amount of influenza antigen vaccine-induced protection does not persist as long as infection-induced immunity. Thus, booster injections may be necessary to maintain protection throughout life. Secondary vaccine failure results if a proper booster immunization are not administered. Many adult vaccines against diseases that are more severe in childhood will not get routine boosters. Although not developing debilitating illness, they may become asymptomatic carriers and infect unvaccinated children. The CDC maintains the most current immunization schedules for people of all ages. Mass vaccination programs have led to major changes in health of the world's population. For example, global eradication of smallpox. By 1979 and 1994, polio was declared officially eradicated in all of America. The goal of the World Health Organization is to eradicate polio worldwide by 2022. Well, I guess we'll have to look that up and see if that happened. Rubella and congenital rubella syndrome were declared eliminated in the Americas in 2015. Excuse me. And in 2016, measles was eradicated. Although eliminated in the Americas, measles and rubella have not been eliminated elsewhere, and infected visitors from other countries may transmit the infection to individuals who have not been vaccinated. A certain percentage of the population, usually about 85%, with a range of 75 to 99%, depending on the particular microorganism, should be immunized in order to achieve protection of the total population, referred to as the herd immunity. The principle of the calculating herd immunity are based on several parameters. This number of susceptible individuals who may become infected in an unvaccinated population from an initial case, the time interval over which the infection may spread, and the effectiveness of the vaccine. An individual infected with measles can infect 12 to 18 other individuals over about 2 weeks. If the desired level of herd immunity is not achieved, outbreaks of an infection can occur. The World Health Organization declared that measles was eradicated in the United States in 2000, and many parents became either complacent or resistant to immunization. This resulted in at least 23 outbreaks of measles in 2014, triggered by exposure to infected visitors to the United States or travel by U.S. citizens to areas where measles was still endemic. Most of the infected individuals had no history of immunization, and the required level of herd immunity had not been maintained. The incidence of pertussis, or whooping cough, in the United States has also reflected resistance to vaccination. In 2004, 32 pertussis outbreaks were published. Analysis of 8 of these found that 59 to 93% of the infected individuals were intentionally not vaccinated. Immunization programs in several European countries have been disrupted by anti-vaccine groups. As a result, the incidence of pertussis increased by 10 to 100 times, compared with neighboring countries that maintained a high incidence of immunization. Resistance to routine vaccination has arisen for many reasons, particularly potential vaccine dangers. As with any medicine, complications can arise, including pain and redness at the injection site, fever, allergic reactions to vaccine ingredients, infection associated with attenuated viruses in immunodeficient individuals, and others. Additionally, some individuals in the United States and Europe became concerned that vaccines, particularly those containing the preservative thimerosal, induced autism in children. Thimerosal is a mercury-containing compound that has been used as a preservative since the 1930s. Multiple studies concluded that neither vaccine nor thimerosal was associated with the onset of autism. The debate related to thimerosal and many other environmental chemicals to autism is still under investigation. Although no case of mercury toxicity has ever been reported secondary to vaccination, thimerosal was removed from all vaccines by 2001, with the exception of inactivated influenza vaccines. Development of successful vaccine depends on many factors. These include characterizing by desired protective immune response, identifying the appropriate antigen to induce that response, immune responses against some antigens on an infectious agent are effective or even increase the risk of infection. Determining the most effective route of administration, injected, oral, or inhaled, optimizing the number and timing of vaccine doses to induce protective immunity in a large proportion of the at-risk population, and deciding the most effective yet safe form in which to administer the vaccine. For instance, most vaccines against viral infections, measles, mumps, rubella, varicella, and yellow fever, contain live viruses that are weakened or attenuated to continue expressing the appropriate antigen but are unable to establish more than a limited and easily controlled infection. The limited proliferation capacity of attenuated live virus appears to afford better long-term protection than using purified viral antigen. Exceptions are the vaccines against hepatitis B, which uses a recombinant viral protein, and hepatitis A, which is an inactivated killed virus. Even attenuated virus can, however, establish life-threatening infections in vaccine recipients whose immune system is congenitally deficient or suppressed. Two different vaccines were developed against polio. The Sabin vaccine is an attenuated virus that is administered orally. It provides systemic protection and induces a secretory immune response to prevent growth of the poliovirus in the intestinal tract. The live attenuated vaccine causes polio in some children who have unsuspected immune deficiencies, 1 in about 2.4 million cases or doses. The Salk vaccine is a completely inactivated virus administered by injection. It induces protective systemic immunity but does not provide adequate secretory immunity. Therefore, even if the individual is protected from systemic infection, the wild type, poliovirus can transiently infect the intestinal mucosa, be shed, and spread to others. For example, when polio was epidemic, the live oral vaccine was preferred. However, about 8 cases of paralytic polio per year in the United States result from the vaccine strain proliferating in individuals with inadequate immune systems. As a result, the CDC currently recommends vaccination with the killed virus. Some bacterial vaccines are killed microorganisms or extracts of bacterial antigens. The pneumococcus is a major case of pneumonia, particularly in the young and adults with compromised lung function secondary to smoking, asthma, or other lung diseases. Of more than the 90 known strains of this microorganism, only a few cause the most severe illness. The current vaccine against pneumococcal pneumonia consists of a mixture of capsular polysaccharides. The conjugated vaccine is recommended for young children in whom the pure polysaccharide vaccine is not immunogenic. A similar vaccine available for H. influenza type B, which is a cause of pneumonia and meningitis in children. Some bacterial diseases are caused by potent bacterial toxins that act locally or systemically. These include diphtheria, cholera, and tetanus. Vaccination against toxins is achieved using toxoids, purified toxins that have chemically detoxified without loss of immunogenicity. Pertussis, whooping cough, vaccine was changed from a killed whole cell vaccine to an acellular vaccine that contained the pertussis toxin and additional bacterial antigens. With so many available vaccines, there has been an effort to mix vaccine in order to minimize the number of required injections. An example is now the DPT, now usually containing diphtheria, tetanus, and toxoids, and acellular pertussis vaccine. A more recent mixture includes DTaP, mixed with an activated poliovirus, hepatitis B vaccine, or H. influenza antigen, or a mixture of these. Coming to our conclusion here is our passive immunotherapy. Passive immunotherapy is a form of countermeasure against pathogens in which performed antibodies are given to the individual. This form of therapy has been used for decades. Horse serum that contained antibodies that was used to treat diphtheria, pneumococcal pneumonia, tetanus, and other diseases in the early 20th century. However, because of foreign proteins in the serum, many individuals developed an immune reaction against the horse proteins and an immune complex-mediated serum sickness. Passive immunotherapy with human immunoglobulin has been approved for several infections, including hepatitis B and hepatitis A. Treatment of potential rabies infection after a bite combines passive and active immunization. The rabies virus proliferates very slowly. Individuals who have been bitten receive a one-time injection with human rabies immunoglobulin to further slow proliferation, further by multiple injections with a killed viral vaccine to induce greater protective immunity. A recent example of passive immunotherapy is the 2014 to 2016 outbreak of Ebola in West Africa, the largest outbreak of Ebola infected ever observed, 28,600 cases, with 11,300 fatalities. No vaccine was available. An experimental mixture of monoclonal antibodies against a virus rapidly increased circulating antibody titers and diminished viral load by more than eightfold. In the past, vaccines and therapeutic antibodies were developed for only the deadliest pathogens, with the increase in antibiotic-resistant microorganisms. The development and widespread use of new vaccines and passive administered antibodies against these microorganisms must be observed and considered. That's it for this chapter. I thank you for listening, for watching, for checking in. But we'll call it a day here, and we'll see you all next time. Thank you, and good night.